Have you ever wondered what gives bacterial cells a structural integrity? Well, it's all about the cell walls, particularly the peptidoglycan layer. The peptidoglycan layer is a mesh-like structure composed primarily of two sugar molecules, N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM. These sugars are linked together through amino acid chains. So let's break down the process of peptidoglycan synthesis into 3 easy steps. The first step is the synthesis of the subunits in the cytoplasm. The subunits are synthesized in the cytoplasm of the bacterial cell. Amino acid chains of 5 amino acids are linked to NAM, forming the precursor subunits. The NAM will then bind to bactoprenol, which is a lipid that plays a crucial role in the transportation of the subunits. And after this attachment to bactoprenol, the NAG will bind to the NAM, hereby completing the first step of the process. The second step of the process is the transportation across the cell membrane. So next, these subunits need to be transported across the cytoplasmic membrane. For this to happen, they are attached to bactoprenol in the cytoplasmic membrane, which has already occurred in the first step. The bactoprenol will then translocate the subunits to the outside of the membrane. At the outside of the membrane, multiple subunits are linked together horizontally by a process called transglycosylation, which is done by transglycosylases. The third step involves the cross-linking by penicillin-binding proteins. Finally, the peptide chains from the glycan proteins are cross-linked by transpeptidase enzymes, also known as penicillin-binding proteins. This process involves a peptide bond exchange, resulting in the formation of a sturdy cell wall consisting of multiple layers of peptidoglycan. This essentially concludes the synthesis, but the synthesis is not always able to take place because there are also certain antibiotics, such as beta-lactam antibiotics, which can come into play. Beta-lactam antibiotics, such as penicillin, contain a distinctive beta-lactam ring in their structure. They target the penicillin-binding proteins, which is responsible for the cross-linking of the peptidoglycan subunits. By binding to these proteins, beta-lactam antibiotics prevent the cross-linking of peptidoglycan subunits, hereby disrupting the cell wall assembly. This activates autolysis, causing the bacterial cell to degrade its own cell wall, ultimately leading to the cell death. And that's how beta-lactam antibiotics interfere with the synthesis of the bacterial cell wall, making them potent weapons against bacterial infections. Thanks for watching. Don't forget to like, share, and subscribe for more fascinating insights into the world of biology.